The overall goal of this procedure is to create a platform for in vitro electrical stimulation of stem cells on a conductive polymer scaffold with subsequent in vivo implantation.
This method can help answer key questions in stroke research and stem cell biology by providing a means to modulate stem cells. The main advantage is that the stem cells can be optimized in vitro and subsequently implanted.
Although this method can provide an insight into stroke recovery, it can also be applied to other systems as a new cell delivery method.
Generally, users new to this technique will struggle due to the precision required for cell chamber assembly and the technique involved with the craniectomy.
In preparation, autoclave the 2.5 by 12.5 cm metal plates and the flow valves. Next, using a chamber slide as a template, cut 2 pieces of PDMS. One piece serves to outline of the chamber's perimeter. On the second piece, cut out 2 adjacent pieces from the inside of the chamber slide. Both pieces should be rectangular and match the chamber slide. Now, layer the materials. Start with the metal plate, then the uncut PDMS, then the polypyrrole plate oriented perpendicularly, then the PDMS with cutouts, and finally the cell chamber. Align the layers carefully and clamp the apparatus together. Next, cut 2 lengths of a metal wire to extend from the apparatus to the outside of the incubator that will contain it. 60 cm lengths are usually long enough. Next, use silver paste to attach a wire to each end of the